Every time a cell divides, it has to duplicate its entire genetic code. The repository for this information is the DNA double helix. It relies on a semi-conservative method of replication, meaning the molecule separates, and each original parent strand acts as a precise template for building a new daughter strand. This duplication has to happen at incredible speeds and with extreme accuracy. To pull it off, the cell must coordinate multiple chemical reactions simultaneously across millions of base pairs. To even access that genetic code, the cell must first break the hydrogen bonds that hold the complementary base pairs together down the center of the molecule. But there is a catch regarding the physical structure of these strands. They possess a strict chemical directionality. One end of the DNA backbone terminates in a phosphate group, known as the 5' end, while the other terminates in a hydroxyl group, called the 3' end. The two strands of the helix are antiparallel, running in opposite directions. This antiparallel geometry poses a massive mechanical problem for the cell when it comes time to copy the data. The primary enzymatic engine responsible for reading the template and building the new DNA strands is called DNA polymerase. This enzyme operates under a strict biochemical constraint. It can only synthesize new DNA by attaching an incoming nucleotide to a free 3' hydroxyl group. Therefore, DNA polymerase is strictly a one-way machine, operating only in the 5' to 3' direction. Herein lies the paradox. The two template strands of the original DNA molecule run in opposite directions, yet the enzyme doing the copying can only move in one. To replicate both strands simultaneously without breaking the laws of chemistry, the cell relies on a sophisticated asymmetric molecular machine known as the replisome. Replication does not start at random locations on the genome. Specific initiator proteins must target and bind to highly recognizable sequences of DNA. This diagram illustrates these starting points, called origins of replication. Notice the highlighted yellow blocks labeled bacterial replication origin. Specific nucleotide sequences unusually rich in adenine and thymine base pairs. Looking down at the yeast replication origin, we see a difference in scale. While a prokaryote like E. coli has a singular origin, eukaryotic cells require multiple origins. Eukaryotic genomes are massive. By possessing multiple origins, known as replicons, the cell can deploy multiple replisomes simultaneously, allowing it to copy a massive amount of data in a manageable timeframe. Identifying these origins is the critical first step. It dictates exactly where the replisome machinery will physically assemble. Once assembled, an enzyme called Helicase acts as a motor protein. It utilizes energy from ATP hydrolysis to break the hydrogen bonds and forcefully separate the two strands of the double helix. As Helicase drives forward, it splits the DNA into a Y-shaped structure known as the replication fork. However, newly separated single strands of DNA are chemically unstable. Left to their own devices, they will naturally attempt to anneal back together. To prevent this, single-strand binding proteins, or SSBPs, quickly coat the exposed strands. This prevents reannealing and protects the vulnerable DNA from being degraded by cellular nucleases. But separating the strands introduces a new physical problem. Forcing the helix apart creates severe positive supercoiling, an intense, twisting physical tension in the DNA tightly coiled just ahead of the replication fork. To solve this, an enzyme called Polymerase moves ahead of the fork. It continuously cuts the DNA backbone to relieve the stress through negative supercoiling and instantly reseals it so the helicase can keep moving. Looking at this schematic of the opened Y junction, we see the two separated template strands. But DNA polymerase III cannot initiate synthesis from scratch. It absolutely requires an existing 3' anchor. To provide that anchor, DNA primase synthesizes a short sequence of RNA directly onto the template, serving as a primer. The left side of the fork here is the leading strand. On this strand, the 3' to 5' orientation of the template perfectly matches the direction the replication fork is opening. DNA polymerase III locks onto that RNA primer and begins synthesizing the complementary strand continuously in the 5' to 3' direction, chasing the helicase as it opens the fork. The leading strand is highly efficient, but it only solves half of the replisome's objective. The opposite strand presents a directional crisis. On the opposite template strand, the orientation is flipped. It runs 5' to 3' relative to the widening fork. Continuous synthesis is chemically impossible here. It would require the polymerase to move in a 3' to 5' direction, which breaks the core rule of nucleotide addition. This is the lagging strand. Because polymerase can only build 5' to 3', synthesis must occur discontinuously, essentially working backwards, away from the replication fork. As helicase opens the fork further, primase must constantly lay down new sequential RNA primers higher up the strand. DNA polymerase then jumps to each new primer, building short, distinct segments of DNA between them. These individual chunks are known as Okazaki fragments. To keep the leading and lagging polymerases from separating, the replosome physically loops the lagging strand template around itself, a mechanism known as the trombone model. This reorients the lagging polymerase so it travels in the exact same physical direction as the leading strand. This complex looping choreography successfully bypasses the chemical limitations of DNA, allowing for the simultaneous replication of antiparallel strands. At this stage, the genetic sequence has been copied, but the lagging strand is structurally compromised. It is littered with RNA primers and physical gaps between the Okazaki fragments. A cleanup phase is required. Specialized enzymes, like DNA polymerase I in prokaryotes, remove the RNA primers and fill the resulting voids with the correct DNA nucleotides. Finally, an enzyme called DNA ligase moves in. Its specific role is to catalyze phosphodiester bonds, welding the adjacent Okazaki fragments together. This final ligation seals the nicks in the backbone, transforming a fragmented assembly line of DNA into a continuous, chemically sound double helix. Because the replosome operates at blistering speeds, occasionally incorporating the wrong nucleotide base pair is a statistical inevitability. When mismatched bases, like an adenine incorrectly paired with a cytosine, fail to hydrogen bond correctly, they create a physical bulge in the geometry of the double helix. If this mismatch is not corrected before the next cell division, it becomes a permanent mutation, altering the genetic code for all subsequent daughter cells. To defend against this, the replosome possesses a built-in proofreading mechanism, an intrinsic 3' to 5' exonuclease activity found directly within the replicative polymerases. When the polymerase senses a structural bulge, it literally shifts into reverse. It acts as an exonuclease to cut out the erroneous nucleotide, inserts the correct base, and then resumes forward synthesis, ensuring near-perfect fidelity. The replosome coordinates helicase unwinding, topoisomerase stress relief, and precise polymerase synthesis into an incredibly efficient molecular machine. The discontinuous synthesis of the lagging strand serves as an elegant evolutionary workaround to the strict chemical directionality of nucleic acids. Looking at this diagram of a eukaryotic chromosome, we can see how this action scales up. At the top, multiple origins of replication open simultaneously. As we move down the tiers, these replication bubbles grow bidirectionally, ultimately fusing together to rapidly duplicate massive, complex genomes. This precise choreography, balancing intense speed, mechanical looping, and active proofreading, guarantees the faithful inheritance of biological information across generations.